people who are designated to throw a buoy out. Method we've been using for about 15 years, and basically what we do is go to the mouth of Winyah Bay, or about halfway down Winyah Bay, and we set long lines, which are about 150 meters long, with floats on each end and anchors on each end, with 25 hooks coming off, baited hooks. They're called gangins, and we soak them for 30 to 45 minutes. They fish on the bottom, or sometimes we'll fish them in the water column or even at the surface. We typically catch anywhere from 0 to as many as 10 sharks per long line. When we do catch a shark, depending on its size, we'll either identify it in the water, measure it in the water, and tag it and release it. If it's 6 feet or less in size, we'll bring it on board. And then we'll take a piece of tissue from the fin, and another piece of tissue as well that we'll put in a library in case anybody needs it for genetic analysis or some other sort of tissue analysis.
Okay, so anchors in, anchor out.
Depending upon what our goals are, we'll either put a tag in its dorsal fin or under its dorsal fin. Lately we've been putting in acoustic telemeters, which are little pingers basically. Make an incision in the abdomen, about a 2-inch incision, and put this AA battery-sized instrument inside. Sew it back up, and it will ping every 60 to 90 seconds. And in order for us to get the information, it has to go by an acoustic receiver somewhere. Fortunately, there's an array of receivers up and down the coast, and indeed worldwide, but the ones we're interested in are in Florida and South Georgia, and then in the mid-Atlantic and the Northeast. And different researchers have put them down, but they all will identify the signature of our unique ping that each shark has, and in that way we'll know where the sharks are.
Here comes our anchor. Last hook, 11:59.
One of our projects is studying juvenile sandbar sharks in Winyah Bay and seeing where they go in Winyah Bay. In Winyah Bay, we have as many as 10 different kinds of sharks at one time. It's a relatively small system. How do they divide the habitat is an important question that we're answering. We're also doing a similar study along the piers of South Carolina, in which we're tagging pier-associated sharks and seeing if they are resident around the piers for any length of time, whether they go between piers, whether it's new sharks coming or the same sharks that are staying there all the time.